Hello and welcome to today's presentation on body defense mechanisms, adaptive immunity. Our focus questions surround what is adaptive immunity, the antibody and specifically the cell-mediated response, and the characteristics of cytotoxic T cells and helper T cells. Adaptive immunity is the response of the immune system to a specific antigen. An antigen is an antibody generator. It is a foreign molecule which stimulates antibody production. The adaptive immune response is specific in that it's able to recognise what is foreign from what is self and has memory qualities in that it retains information and helper cells to rapidly fight future infection. The adaptive immune response involves white blood cells, the B cells and T cells, and there are two types of responses. Displayed on the left-hand side of the diagram is the antibody response, which utilises B cells, and on the right-hand side of the diagram is the cell-mediated response, and our focus today of T cells. T cells come in two different types. The cytotoxic T cell, displayed on the left-hand side, associate with the MHC class I molecule. They are directly responsible for the breakdown and destruction of the foreign antigen. The helper T cell, displayed on the right-hand side, associate with MHC class II molecules. They assist the immune response of other lymphocytes by secreting cytokines such as interleukin-2, which is the co-stimulator for the activation of other helper T cells and cytotoxic T cells. The activation of our T cells comes in three main phases, and we can see this in the diagram. Firstly, they recognise the foreign antigen at the plasma membrane of the infected body cell. The antigen has previously been broken down and inserted into the plasma membrane by the help of the MHC class I or II molecule. In this diagram, we can see that it's MHC class I because it is associating with the cytotoxic T cell. The second phase occurring is called co-stimulation, which is aided by helper T cells and interleukin-2, and it's the process which maintains the coupling between the antigen and the now activated T cell. Finally, now that our T cell is activated, it begins to form clones of itself to swiftly destroy the antigen, which is a process called clonal selection, in which the T cells proliferate and differentiate into either active T cells, which attack the infected cell, or the memory T cells, which remain in the body for the future recognition and rapid immune response. And lastly, our cytotoxic T cells destroy foreign antigens by binding to the infected cell and undergoing either one of two processes. Displayed on the left is shown the release of the granzymes, which break down the infected cell, allowing it to then be destroyed by phagocytes. Or is the second process displayed on the right-hand side is the release of periphonin and granulizing proteins from the granules, which break down and kill the cell directly.